John, we can start whenever you're ready.
Okay. Well, welcome everybody. My name is John Bates, and I'm from the Field Museum, where I'm a curator of birds, but I'm also a member of BCON, the Natural Science Collections Alliance, and the American Institute for Biological Sciences Board. Um, you're here today as part of our One Health listening session, which is part of the BIOFAIR data network. And that's about building an integrated, open, findable, accessible, interoperable, and repeatable uh, uh, data network. And so the first thing I want to do is do a little housekeeping. I want to emphasize to people that this session is being recorded, and we're also going to use some AI summary tools for note-taking purposes. We know that particularly folks from the federal agencies sometimes have some issues with this. If there's somebody who has objections, please let us know in the chat, and we'll work on this. So we also hope you'll engage with us with the camera on, just because we can see folks. We encourage you to use the chat feature to share comments, ask questions. We'd also like you to introduce yourselves with your name and your institutional affiliation, just so folks can have an easy access to seeing who's here. If you have any issues, you can contact AIBS IT support via the chat, and Diane will help you out. And then today, the agenda is, this is a listening session, so there'll be several presentations that'll last for about half an hour, and the remainder will be dedicated to discussion. I mean, the idea is we want to hear from you all as part of the One Health community. So we are going to address questions after the presentations. There'll be a chance to do that. As we're getting into the discussion, if you can raise your hand in the chat, via the response tab. That'll be easier because we'll be able to see who's trying to get into the conversation. We're going to have a breakout session, and there are going to be opportunities there to type into a Zoom chat or into a Google Doc, but you can also use the chat at that point. Organizers will be taking notes, and that's for a summary for the report. So I just jumped forward. I did. So the Biodiversity Collections Network is a group that was organized around, based on an NSF grant a number of years ago, a research coordination network grant. And the idea was to bring together the biological collections community to do a better job of integration associated with understanding and promoting and conserving the diversity of life using the data that are in museum collections. And we want to promote the accessibility to these collections and their impacts that they have for biodiversity, and get them recognized as a critical part of the infrastructure for research and education associated with biodiversity. So the BCON Steering Committee is a group of folks that come together on a regular basis. We represent a broad spectrum of the traditional museum community, but we also include paleontologists, cultural collections. And if you're interested in getting involved with this, stay in touch, because we're always interested in broadening the capacity that we have to represent biodiversity data writ large. So BCON activities have, over the years, included advocacy, community surveys, podcasts, webinars, workshops, and publications. From a publication standpoint, we put out the Extended Specimen Report, which I'll talk about in a few minutes. But we've also done a number of peer-reviewed articles, the most recent one advocating for specimen management plans in National Science Foundation proposal guidelines. And so the ideas that we have out there is again, to promote the science done in the collections community for use in all kinds of different types of science. So I've mentioned this already, but some of our current initiatives include the Extended Specimen Network and the Specimen Management Plan, but we're also involved in education efforts such as the biodiversity literacy and undergraduate education, or BLUE, which was started through Beacon and is run by Anna Monfields out of Central Michigan. And then from the advocacy standpoint, we've been involved in the reports that we've written, advocating with the Office of Science and Technology of the President and other government entities. And actually, we made some museum collections were in the Chips and Science Act, which is an incredible breakthrough. And of course, the result of that has not been manifested in terms of money being applied to this yet, but those are the kinds of things that we need to do for our communities moving forward. So here are a couple of publications that came out that led to the development of and support from the Office of Science and Technology and the President and the Chips and Science Bill. In 2017, a group came together across the community to discuss how we could implement this extended specimen concept, and resulted in a report that was sponsored by the National Science Foundation. And then there was an accompanying article in Bioscience that did a less technical description of the concept. Basically, what we're interested in is taking advantage of the digital world that we find ourselves in. So we want these specimens to be born digital going forward, such that they can be used in all kinds of different ways for different types of science. At the same time, we are painfully aware that a lot of specimen digitization, specifically in hyper-diverse groups of organisms, is still lagging behind, and we need to follow up and getting more specimen digitization in groups like insects and invertebrates. Going forward, we want to see more data integration and cyber infrastructure, and that involves making community and institutional infrastructure stronger too. And then along the way, we want to create a stronger and more diverse workforce as we go forward. So extended specimens rely on data integration, which is one of the reasons why we're doing these listening sessions. For One Health is a perfect example of that, and I hope we'll be able to talk to a broader spectrum of the community in One Health in our discussions today. But the collections community, as you're going to hear from people in that community, represent a huge opportunity with respect to One Health data, and we want to see that developed further using these FAIR data concepts. So the schedule for the grant that we have is, we did some training associated with developing mechanisms for scientific communication on the topic in the spring of this year. And now we're doing the listening sessions. This is the third of 6 listening sessions that are coming up. And then in 2025, following the listening sessions, we'll compile the data and put together a 2-day virtual workshop, which will be a broader spectrum of people from the community, but be limited to about 60 people or so. And then by the spring and summer, we'll put together a synthesis with a final report and a number other of outcomes describing where we see this data network going. So here's that again, one more time, just showing you the directions we see with the final output of the BIOFAIR project. And we want to encourage folks to participate in this. I want to emphasize that in addition to the traditional overarching groups that you often hear about, such as AIBS and Spinach and the Natural Science Collections Alliance, we're working with the data folks such as iDigBio, the Biodiversity Standards Group at Tadwig. And then again, we're very cognizant that one of the things that needs to happen is we need a more diverse and representative community across the board. And how we do that is through interactions with groups like Blacks in Genetics. And we're always looking for other opportunities to expand participation in this effort. So here are these 6 listening sessions. We call them domain focused. We've already had federal agencies and genetic and genomic data. Today we're doing One Health, and then you can see the other 3 that still remain to happen. If there'll be an expression of interest form that will be linked to in the chat, and you can, if you'd like to come to some of the other sessions, we'd encourage you to do this. And so there's a summary on the right there of the discussions that we hope to have across each one of these listening sessions. And so for the One Health session, I just want to make sure I introduce the people on our group. So this was led by Gregory Watkins Colwell, Julia Portman, and Sheila Poo. And then our key domain representatives today who are going to take over from here include Kelly Spear from the collection of pathogens and parasites at the University of Michigan, Marcy Ravelas from the CDC, and she's in charge of frozen cryogenic collections at the CDC. And then Joe Cook from the Museum of Southwestern Biology and the University of Michigan, who's been a longtime researcher working on lots of different questions related to One Health type data. And so with that, I will introduce Kelly and let her take over and talk about some brainstorming that we wanted to try to do.
Thanks very much, John. Would you be able to share the brainstorming notes in the chat? Um, so just to get us started, we wanted to brainstorm together who we all envision being the major stakeholders in this type of One Health network that is inclusive of natural history collections. So John is sharing on the screen our outline for who we imagine to be the major players right now, but there's space to include others we haven't thought of. So I'd encourage you to raise your hand, turn on your video, let us know who you think those major players would be, or share in the chat your ideas. I might start doing my start wearing my instructor cap and calling on people. Tim, you're coming from a really unique perspective as like a fungal collection. Who would be those major players in the world of conservation for chytrid?
Um, well, there's hi everyone. There's partnership for reptile conservation and amphibian conservation. Those are one. And so I think that's PARC, Partners for Amphibian and Reptile Conservation.
Is that an NGO?
Yeah, I believe they are an NGO. And well, there's the federal agency on the USGS. I think that's going on there now as well. Well, that's populating fast. So there's a couple ideas there relevant to the amphibian disease.
Thanks, Tim. Yeah, that's perfect. We hadn't had NGOs on our list yet. I see you folks populating our Google Doc. Laboratories, these, Great Point, government labs. Um, Anyone else have ideas about NGOs that are really important partners in this One Health realm? I'm seeing the Nature Conservancy, Audubon Society. Yeah, all great. For the NAHLN, I'm not familiar with that acronym under labs. Can whoever put that in there tell me more about that?
Oh, that was me. Hi, Kelly. I'm Jocelyn Colella. I'm at the University of Kansas, curator of mammals and assistant professor. The NALN Labs are the National Animal Health Laboratory Network, which does a bunch of diagnostics and ongoing disease surveillance. And I think particularly digitally linking material or vouchering material back to museums would be especially helpful in that realm.
Thanks, Jossie. Yeah, great point. Great. I see that someone created a new column to create a new category of stakeholder that we don't have yet, but hasn't been filled in. Um, can whoever created the new column tell us more about their plan?
Oh, that was me, Jyotsna Pandey from AIBS. I just created it just in case people thought of another category
Oh,
in
thanks.
anticipation of other.
Great, thank you. Yeah, we are missing maybe some community engagement components in this, although I know each of these major categories do a lot of community engagement on their own. I see advocacy being filled in. Great point. Should we add in anything there? We'll wrap this up in about the next 1 or 2 minutes, so get your thoughts in. Yep, MEPA, seeing communities of practice going under advocacy. Great. So we'll wrap this up so that we can move on to the listening sessions, but Paloma, I see Wildlife Conservation Society, we can add that to the NGO list. So thank you so much for sharing your thoughts. I just wanted to get us some practice with getting our thoughts on these shared Google documents, but as we go through the presentations, you'll still have access to this document and you'll be able to add to these thoughts as we go through the slides. John, would you mind sharing
Yeah,
your slides
I'm
again?
I'm absolutely trying to figure out how to do that.
Sorry. I think it's that second tab there. Higher up on your screen.
Uh,
Yes. That yellow one. One
got
Health
it. No,
BioFAIR.
it was covered up by the reactions tab thing. There we go.
Thank you for doing that. Okay, so to get us started, I'm just going to give a quick introduction to what the extended specimen is. A lot of us are already familiar with this term, but we may have different understandings or different perspectives about how to apply that extended specimen so that we can approach our discussion sessions later from the same foundational understanding. I wanted to have these few introduction slides. So uh, if we back all the way up to the central tenets of the scientific process, at its core, science is really reliant on the processes of validation and replication to ensure the research that we're doing, the conclusions we're reaching are reliable and as close to the truth as we can get. So this validation step and this replication step are centrally hinged on the ability to revisit specimens in a museum collection from the perspective of One Health. For example, these museum specimens, which can be whole organisms or samples from organisms, these can help us do things like confirm a host identity. So if we're trying to understand the reservoir hosts of a specific pathogen of concern, having this catalog of those host specimens to confirm that host pathogen interaction is an absolute necessity, especially as we're moving through, or especially as we're updating the systematics of these hosts. It also allows us to revisit specimens and test them again for additional pathogens. For example, with the emergence of SARS-CoV-2, if all of the bat specimens from which coronaviruses in China had been sampled were put into a museum collection and made available for additional study, we could revisit those specimens, re-examine them for coronaviruses, and have a better understanding of the potential spillover route of this particularly harmful virus. Essentially, these museum specimens offer a nexus for extension. Not only can we understand some central components of the biology and ecology and evolution of the host, but it also allows us to extend our knowledge and understanding of host pathogen interactions in immunology and in One Health. This is important in a One Health discussion with an audience that includes museum professionals and also folks with different expertise, because This permanent specimen step is often missed depending on the traditions of your field. For example, virology, this is not typically part of the process that happens once you sample, depositing those samples in a museum collection. 30 of 31 published beta coronaviruses and 95 of the 139 distinct hantaviruses cannot be traced to permanently archived samples. So that limits our ability to do this validation and replication step. So what is this extended specimen? So each specimen represents a nexus of multiple types of information. For example, take this mouse image. You can derive measurements of that mouse, tissues for further genomic or transcriptomic analysis, ectoparasites, spatiotemporal georeference data, microbiome, virus, and bacteria data, endoparasites. We record ecological field notes, morphological components, and also high resolution images. So together, this suite of data is known as the extended specimen. We have the host vouchered organism, and then all of this additional information that's valuable for One Health that we can apply to the types of questions that are illustrated on the right. So related to community structure, interactions, pathogen, ecology and evolution, host ecology and evolution, etc. Next slide, John.
Yep. Why is it not?
Thanks. Oh.
Yeah. Good help. It's hard to find.
No, this is great. I appreciate it. So essentially the take-home message is that, museum specimens and their associated cryo collections are resources of high quality data. I'm here representing the Michigan Pathogen Biorepository. Joe Cook is here representing the Museum of Southwestern Biology. We also have Marcy Reveles, who is representing the CDC's biorepository. All three of these biorepositories are seeking to accession these high quality wildlife specimen data to make them valuable for One Health or extended specimen purposes. And we're trying to find ways to make those specimens more accessible, more discoverable, more usable in that One Health realm. One of the major initiatives that our cryo collections have recently undertaken is this idea of the global museum, where we try and implement locally anchored infrastructure, essentially extending the tent, extending that infrastructure and access broadly. So one of those initiatives is through a consortium called BioArc, where we're attempting to build biorepositories, so ultra low freezers across Latin America at these locations with partners locally. These biorepositories would store wildlife samples in ultra cold freezers backed up with generators and make the data of these wildlife specimens available through an Arctos database that can be shared not only with other scientists and scientific platforms, but also make it easier for communities to engage with this type of sampling process happening in their backyards. Arctos database can connect with web-based apps that enable community scientists to help record information relevant for One Health and pandemic preparedness locally. Essentially, we hope that these biorepositories and natural history collections can help us understand these determinants of spillover. So if we consider this kind of seminal pyramid of spillover to pandemic, the wildlife samples that we're collecting are going to enable us to understand the reservoir host distribution and density, pathogen prevalence, infection intensity, and pathogen release from a reservoir, which are those determinants that make a pathogen able to move from enzootic circulation to a spillover event. Then in combination with climate data and other high quality data, we can also use these high quality wildlife specimens to understand the downstream steps that move from outbreak to epidemic to pandemic. And I'll turn it over to Marcy now.
Hey everyone, can you hear me okay? Okay, great. So just a couple of slides and just to kind of, I don't know if this is really flowing quite well with what Kelly's been talking about, but we've come on board and we're very interested in hearing and participating in this initiative for sure. As mentioned earlier, I do manage the CDC biorepository, and right now we really are kind of come a long way in the last few years, just trying to modernize this facility. It's really sort of been kind of in the background of just being a long-term storage for a lot of legacy samples that have been collected over time through the agency. These samples are considered valuable, of course, otherwise they wouldn't be archived, and we do provide ongoing support to CDC programs. There's dozens of programs and labs across the agency doing very different things, and it's an interesting place to be right now, and this kind of initiative really fits well. Right now, the biorepository houses about 7 million samples, and we do play a crucial role with the agency in terms of public health research and population health by facilitating scientific knowledge advancement, characterization, for example, of new etiologic agents, investigation of disease causes, the development of new tests, assays, vaccines, treatments, and so forth. But with respect to any kind of data infrastructure specific to these samples, we've been working kind of retroactively to get things digitized. We have the same kind of challenges that natural history museums face in terms of, with legacy collections, getting all of the available data and information associated. Right now, all of the samples are really only available internally to the CDC. It's hard right now to get things external in terms of access, but we are working toward that, and certainly being involved with something like this is important. Most of our samples, though, right now are human, they're not animal, and I wanted to emphasize that. A lot of the samples that are generated, stored, tested even by the agency really don't even make it to our facility at times. They're consumed in the testing process, but use of our facility is voluntary, and not all programs seek us out, but we have the ability to do that. Programs across the agency would collaborate with researchers like yourselves or museums, of course, and samples could end up here. We aren't necessarily limited to just human samples. It just hasn't been a focus. But again, I just want to say I'm here mostly today to listen. If any of you have ideas or already are working with collaborators, please let me know. CDC is a rather large, complex organization, thousands of people working, but I'm here to help as much as I can and help facilitate. I do want to point out this image here. It's not unheard of. Similarly, as a natural history community, these legacy collections don't just sit there and get unused. And a study in 2016 was completely looking at something different, but stumbled across these old legacy samples. I think they were from the 1970s, but when they were tested, HIV popped up, and it really rewrote the timeline and what we knew about HIV and how it entered and spread across the United States. And you guys talk about patient zero, and that's oftentimes used a lot now in different scenarios, but that sort of originated with the HIV situation with the guy who got a really bad rap for having sort of introduced that. Turned out not to be him at all. So there's a lot of interesting things that can happen with samples that have been stored with us over time. Next slide, John. One of the ways that I can also facilitate is not just being part of the biorepository, but I'm also chair right now, CDC Sample Policy Board. I think this would be a great group for us to sort of socialize this, possibly to the agency through a presentation or what have you. But there's a lot of different people who are sort of senior stakeholders and begin conversations of how we can better service and collaborate with the natural history community. Certainly having come from this group, it's certainly a dream to bridge that really quite nicely and set up, oh, the lights just went off. Set up a roadmap to increase access and address data standards. There are policies here at the agency that deal with sample management and data, and some of these are in the current state of being revisited and revised, and so these conversations can certainly play a role in how those are drawn up in the future. And that's it for me. So I don't know who's next, but thank you for this little opportunity to pitch.
Yeah, hi, I'm Joe Cook. I'm going to tell us a little bit about a community of practice and the idea that communication across disciplines is super important for One Health, and that building on this idea that Kelly introduced, that specimens are a nexus for many different disciplines. And so the idea that this may be a really good forum for bringing together people that are approaching One Health from different directions. So today I'll talk a little bit about MEPA, which is this group that we spun up right after SARS-CoV-2 emerged, called Museums and Emerging Pathogens in the Americas. The idea was to start conversations between the biorepositories in Latin America and in the US with public health officials and pathologists and wildlife biologists and so on and so forth throughout the Americas. The idea of these community of practices spun out of Project ECHO, which actually originated in the biomedical field. So this is an example of museums actually riffing off of something that was developed in the biomedical field. Actually here at the University of New Mexico, originally started to look at hepatitis in humans in rural situations in New Mexico. It quickly led to a network that expanded through Chihuahua and Sonora, as there was a lot of movement back and forth across the border, basically to get information from rural communities about what was happening out in those instances, but also to give information to those communities with regard to hepatitis. So this is an idea about networking, but definitely trying to bring different disciplines together to develop new perspectives and also diversify collaborations. With regard to MEPA, we hold a bi-weekly discussion where the format is an hour, where we meet every other week with a 30-minute presentation and then 30 minutes of discussion, with a real focus on the discussion to get people to throw out new ideas and find new ways forward. We've also talked a lot about visioning next-gen collections in this. This is the example of the distribution of some of the participants in this and the key players. Jocelyn Colella has already spoken to you today. Cody Thompson is also on this call today, University of Michigan, and then a number of partners in Latin America, including Hernani Oliveri here and Alejandra Camacho, Alejandro in Ecuador and Hernani in Brazil. Next slide, please. As I mentioned, it's affiliated with Project ECHO. This MEPA now has about 270 people that have participated, including 175 professionals and 99 students. We have a real focus on capacity development, so bringing students into the discussion early on to learn about how multidisciplinary approaches can be useful in One Health, and participants from 16 countries. Next slide. So we talk about what are the key questions in One Health, and what are the key questions for museum in terms of their role in One Health. One of the questions we spent a lot of time thinking about is how can we sample strategically across scales? And this is the idea that in the museum community, a lot has been made about the extended specimen, but unfortunately, collections have not been built in the past to address One Health issues. And so a lot of the extended specimen discussion has been about sort of serendipitous use of specimens rather than strategically building this infrastructure so that it will be useful for One Health efforts in the future. And so We have been introduced today to the holistic or intentional extended specimen instead of the serendipitous extended specimen. We talked a lot about when and how often to sample across time and across space, because collections are really good at helping us with scaling issues, and also across taxonomic diversity. So what species should be sampled? Should they be keystone species, endangered species, commensals? How do we sample symbiotic relationships? Should we be getting whole community snapshots at particular times and places so that we can understand interactions better? And then what about interactions among pathogens in a particular host? So how are the bacteria, viruses, and helminths, for example, interacting? And then other considerations as well. Next slide, please. So again, just to reinforce the need to sample strategically, we could use a lot of help in thinking about this from other disciplines, from folks that are professionals in virology, helminthology, and other fields. The idea that the specimen can form a nexus for a variety of different kinds of study, really giving us a complete One Health view. And also, we talk a lot in MEPA about the need to build sustainable infrastructure, particularly in the global south. And this was Kelly's point about locally anchored infrastructure. That includes building human capacity in terms of curatorial staff, data scientists, and educators, but also buildings, cases, nitrogen freezers, and associated informatics resources, because there are a ton of data associated with every single specimen, which are georeferenced and have a timestamp as well. And then finally, of course, we need to do policy engagement, think about how we might help shape Nagoya as we move forward. And of course, engaging and benefiting local communities underlies the whole process. Next slide. Okay, John, you're on.
All right. Well, we're going to get into the discussion here, but we'd like to encourage folks to ask any questions that came up as the result of the presentation so far. Anybody?
I have a question. Sorry, I updated Zoom and now I can't figure out how to raise my hand.
No problem.
This is Kendra Phelps. I'm from EcoHealth Alliance, which is a nonprofit that I guess goal is pandemic prevention, is their motto. One thing, so I presented last week at the American Society for Virology, and my whole discussion was about how virologists in particular, if they're going to do disease surveillance work with wildlife, how they can maximize their money, time, and effort. One of those was vouchering specimens. One question I got was whether museums can handle BSL-3, BSL-4 level pathogen samples.
Let's see. We've got some experts on that. Joe, you want to make a comment on that?
Yeah, BSL-3, we can handle. BSL-4, we have not dealt with. And I think Kelly and had some discussions at the University of Michigan about this as well, right?
Yeah, it seems like, yes, a lot of museums I know, also Jocelyn Colella's collection are moving towards being able to handle BSL-3, but BSL-4 might be, given the distribution of museums across the US, it seems like, and the rarity of BSL-4 labs, with good reason, potentially there's opportunities to partner with those labs to create satellite collections in those BSL-4 spaces. But I don't know of a museum collection that has BSL-4 access right now.
Marcy, do you want to comment on that too?
I can. Having been here for a while now at CDC, I kind of cringe at thinking of VSL 3 and 4 storage conditions and what that means now. But I'd like to say that, given the right situation, a place like CDC could host that, but our space is limited as well for those activities. But, if anyone's interested in learning what those requirements are, what needs to be done, we can certainly help have those conversations for sure. For us here at the biorepository, we are outfitted in the laboratory to manipulate and handle samples only at a BSL-2. And we can store samples at a 3, but the requirements and the hazards really jump up from a 3 to a 4 when you're opening, thawing, and handling the samples. So I just want to express caution, making those distinctions of activities with what you do with the samples or the specimens.
Anyone else want to weigh in? Other questions?
I guess would it be possible to save some of the biological material in a smaller amount, like nucleic acids, for example, when you're dealing with stuff that has potentially 4 agents in there, so they're somewhat deactivated, and then some other types of deactivation for said rodent or organism for that matter?
That could be a possibility. Oftentimes, though, especially with BSL-4 pathogens and BSL-3 even, they want to culture those particularly viruses. So deactivation would not be ideal, but it's something that can be done for like the whole animal. So after you've pulled your tissue of interest or your diagnostic samples, then you could treat at least the voucher specimen, the whole organism, in a way to inactivate most pathogens. I'm just thinking of like diagnostic samples or tissues that are pulled from an animal, then sent to the museum, swabs, let's say, are tested, and then they realize this was infected with like Ebola Zaire. And now the museum has got specimens and maybe other tissues that are not inactivated properly. And just if there's any museums that are prepared to handle that.
Yeah, these are conversations that we've been having more and more frequently. The way I'm curious to hear from other folks, what within, it seems like there's a within each institution how folks are handling this, but for our pathogen biorepository, which currently is BSL-2, so anything above that we have to inactivate. If something is discovered in one of our, flash frozen tissues, which could have live infectious virus, that is like a select agent or higher than our BSL-2 capacity, we are required to inform the CDC and inform our EHS and then potentially destroy those specimens or otherwise inactivate them through like formalin fixation. That's our protocol right now.
Yeah, Bridget, go ahead.
Yeah, so I mean, I primarily work with fungi, but there are, and BSL-3 fungus in particular. I think it's the same problem that clinical labs have, and most clinical labs also don't have BSL-3 or BSL-4. So just having a network that, so for example, a Phoenix lab finds out that they have anthrax or burkholderia or something in diagnosing a patient. We've set up sort of a network that we can go and retrieve that sample and bring it back to our BSL-3 to process that sample. We don't have a BSL-4, but we can at least deal with select agents at a BSL-3. So perhaps thinking about networks where if that is discovered, because oftentimes, yeah, the default is destroy. And then we lose that, we lose that piece of history, which is the purpose of trying to do this work. So I don't know if that's a potential option.
Yeah, I certainly think that is. Any other questions, comments? All right, I'm going to share my screen again here, assuming I do this right.
Oh, we have another comment from Sabrina.
Oh, great. Thanks. Where is that? Am I not seeing that?
Hi, it's Sabrina. I don't want to hold up the program, but if there's still time for questions, I have one that's sort of outside my area of work, but I think is relevant to this discussion. And the question is about storage and cataloging of samples from domestic or livestock animals. And as we're seeing when it comes to spillover and the emergence of disease, these domesticated animals or agricultural spillover events are kind of increasingly part of the picture. And so I was just wondering to what extent there's been a discussion among people who work more directly with collections about whether it's now appropriate to be cataloging domestic animal samples in some way. Is that something that the USDA is involved in? Is that I feel like it's something that we don't think about maybe as much as we could.
Yeah, great question. I think historically, natural history museums have not dealt with domesticated or even commensal organisms. So commensals like mus and ratus, right? But we recognize that this is an important missing piece of understanding One Health for sure. We haven't dealt too much with livestock, but we are certainly increasing our collections with regard to mus and ratus and other mammals that are closely associated with wild mammals, I guess, that are closely associated with human dwellings and humans. And you've identified certainly a need, and maybe USDA would be interested in helping with that a little bit. We do have some cattle collections that are associated with a very large bison archive that we have related to brucellosis, but related mostly to sort of understanding hybridization between bison and cows.
Yeah, I would just say on a much smaller scale, we've made a point in international field expeditions to collect chickens and geese on occasion just for bird related research, just because we recognize that there's absolutely some connectivity there that's worth having some information about. Anybody else? All right, I'm gonna turn it back over to Kelly here. And go ahead.
Okay, great. I think Marcy is going to be helping me moderate this section as well. And John, feel free to everyone feel free to jump in. So, okay, so we're going to lay out 3 different kind of discussion topics to help us get towards this, developing some ideas about how to make our current system more robust. So for our first discussion topic, we're going to consider the example of the emergence of hantavirus. Just laying out a few facts here. For 30 years, we thought hantaviruses were solely rodent-borne and highly host-specific, but museum collections have revealed a much broader host range and geographic distribution for these viruses. Museum collections were used to help identify deer mice as important reservoirs of the 1993 hantavirus outbreak in the southwestern US, and continue to be used to help discover previously unknown reservoirs of hantaviruses, for example, bats. Collections allow us to look at host pathogen interactions spillovers, and emergences in ways that would otherwise be impossible. So given this little tidbit of information, would this One Health approach, inclusive of museums, have made our responses to other emerging zoonoses more effective? So given hantavirus as a model for an excellent integrative approach to One Health that includes museums, what can we learn from this exemplar to make our responses to other emerging zoonoses more effective? And we will be keeping notes in the chat and summarizing those notes at the end.
Well, if we wanted to do just out there ideas, unlimited funds would be like metagenomics of every specimen in every museum. That would give us a clue on a lot of the unknown or novel pathogens that are potentially out there. Obviously, there's subsequent testing to see if they would even infect human cells. But I'm sure in someone's collection there is something we don't know about that potentially could be pathogenic. So given unlimited funds, unlimited staff and expertise would, more screening of the voucher specimens that are already in holdings.
Yeah, I love that, Kendra, the idea.
I also agree to that.
Go ahead, Rick.
I agree. You're here, Kendra.
Thanks, Ray.
So I guess, Kendra, that's like the broad idea that museums can help us be more predictive. So rather than wait for the emergence of a new virus that's harmful.
I think it's we don't know what we don't know, right? But the problem is when you go up for traditional funding, they're like, well, what are you looking for? I'm like, I don't know. I'm looking for disease X. And they're like, well, we're scared of disease X, but we don't want to fund anything related to disease X because we don't know what it is, right? And so I think, we have to do a surveying to see and monitoring. We can't do any of the, we don't know what we don't know. Sorry.
No, along those same lines, Richard, I'm always fascinated that when you get the ebb and flows of disease outbreaks, testing tends to stop once the whatever's out there subsides. And West Nile to me is a perfect example of that. I don't think we have a whole lot of ideas about how West Nile is circulating out in the natural world. And can we identify strategies to actually get on top of some of those questions?
Johnny, John, I completely agree with you. West Nile, we don't even have high quality data to know where it is, and there's so much disparaging and like collection of the data with just even simple latitude, longitude of where cases would be in insects or mammals or humans would be absolutely superb, right? And so, we can't model something that we don't know that it's out there, right? And so, but normally what we get is, well, it's either not a problem yet, and therefore we do not care, or it was a problem, but it's no longer a problem anymore, or it's not a hot button issue now. But I think that is at detriment to all of us.
So how, given the scenario that Kendra laid out, we have unlimited funds. We're going to metagenomic sequence everything we have in museums, and we're not going to be worried about, funding. We're not going to worry about, all of that stuff. Who would be involved in that sequencing? Like, what would be that network we would want to ideally see? And who would we share the data with? How would we make that metagenomic sequencing effective?
I think a lot of us, at least we did in Charlotte, we built up a lot of infrastructure during COVID to do routine COVID sequencing. I have an nanopore here that can do 24 flow cells a day and produce 1.5 terabytes per flow cell. We built all this infrastructure with COVID, and yet we're just all going to stop now, right? When could be something that we could be monitoring now. And so I think that, obviously we're continually doing wastewater, which I think is important, but I think we should also be looking at the natural reservoirs, and that's why I'm here, right? And so it is, I think, in my opinion, the funding to get people's head around that we need to be proactive and not reactive.
Sure, I hear you, Rick. Nikte, yeah.
Yes, so on the same idea, I mean, it's important if we could do like museumics and have all that information, but as Richard said, it's important to keep collecting because all these will be evolving. So we probably we'll have an historical background with museums, but it's important to say that we need to keep collecting and also use like new tools like artificial intelligence tools, because that could give us like the potential of pathogens that we are not finding now. So it's important to combine both historical collections, collecting in the present, and also if we have limited models, because we don't know, like models will have a limit, but if we can improve with these tools, I think it will be important. And then of course, sharing to everyone, like have our museums or like what we do now with the specimens at museums, we have this net and we say, okay, we have collecting this and that, and then we have these websites for genes or genomes, but we can also have these public access with these new potential pathogens.
Yes, absolutely. And our part 3 discussion will talk a lot about how to be strategic about that future sampling that museums can do. So it's a great point. So, okay, we've heard that, yes, we need to do future collecting. We need to continue collecting, not just rely on historical collections. We need to secure that funding to enable us to do this screening. But let's say we have these data. Who would be the people that we would want to share this data with to make it effective in a One Health context? How can we make our current approach, where we're kind of each doing our own little surveillance projects, more effective?
So I think, the way that we did it for the human microbiome projects and these type of things, we just made it publicly available and easy to search. So then that allowed people to get the data and have robust metadata and standardization there, where people could reuse the data over and over and over again. So I just think getting it in the hands of folks that are computational and having it potentially in multiple different locations allows it to be more usable. And NIH is great about this, and I know NSF as well is great about this with like Neon, for example, but putting them in formats that are easily accessible, even potentially tokenizing certain data formats to be easily used in stuff like machine learning, I think are initial valid ways. And even just having like what they have for Neon, where you can just go and grab it.
I'm seeing a couple of comments in the chat. Bridget and Jennifer, would you feel comfortable sharing more about your thoughts?
I can, sure. I mean, some of these diseases are going to be reportable according to CDC. And so I think maybe that's something that wouldn't be a habit for people to potentially report these from museum specimens, but certainly communicating that information. I think having like the Neon database type format for the data in general would be really helpful, but also thinking in terms of public health interventions. Certainly that whatever state you're in, there would be public health officials that should be notified. And then depending on the status of that, up to CDC.
This is Jennifer. Yeah, I was kind of thinking of the typical, wildlife, livestock, human interface, which veterinarians are usually kind of right at the forefront of a lot of that. And whenever, that's where 70-80% of pandemics start with a animal pathogen. So having access to that data that could help expedite learning where that disease comes from and how it is mutating and how it is transmitting could really speed up the process of mitigation.
Yeah, great. Thank you so much for sharing. Are there other thoughts before we wrap up this part 1 discussion and move on to part 2?
I wanted to ask Richard a question, which is that I'd be really fascinated in how good the use statistics are when you make those data publicly available. So can you actually keep track of how people are using those data and build from that?
Absolutely. And there's such good reporting things, especially in stuff like, even if you have the software associated with moving it, for example, that we can take track of, and we get very, very good analytics for this kind of things. And obviously, the end result is it gets cited, right? Like that's the thing, is like when you get a citation, then to the Hallmark paper, for example, of whatever that data that's generated, it really shows you. So, and I think HMP did really well with that, right? Upwards of most of them have over 2,000 citations, right? So I think we can have, and NSF and NIH both love to see that, right? And I imagine DoD, NASA, they're like, okay, well, you generated all this data. And I think that was the thing with HMP, where it's just like, okay, we generated all the data. Now we obviously get the Hallmark papers, and then here are the analytics on how many people are developing hypotheses and stuff around that. And I think, the classical funding agency, NSF, NIH, like that. And I think DoD is also kind of making a push in that area, stuff like Army, Office of Naval Research, those type of things as well. And so I think that's where they're headed in their minds. And so, yes, we can make that available, no problem.
Cody, do you want to?
Yeah, no, I just thinking about this question, I guess, from the perspective of the timeline that, the hantavirus outbreak happened in the early 90s to where we are today in the post-COVID world. Yeah, I think one thing that I know Kelly and Tim and others have experienced is that, museums were not accustomed to this, high biosafety sort of pathway of doing things. And, there certainly are people on this call that kind of straddle both worlds. And I do think that's, at least from a university museum perspective, that our health and safety people are very risk averse. So, and one thing I think museums are going to have to figure out a way to be able to continue to do our bread and butter, our systematics and taxonomic work that we've done for centuries, and think outside the box. And I think what Nick Tay mentioned previously about continuing to grow. I mean, one thing that I think anybody has done field work in the past couple of years, I mean, really the past couple of decades, but especially more recently, it's become really hard to do anything, even domestically within the U.S. And we really need to educate, I think, our agencies, both at the local and federal level, as to why these continuing collections are important, but also realize that there is some issues with the risk that they're going to have to hopefully tolerate. I think I don't know how to better way to put it, but I think for example, working with hantavirus and Joe's backyard, working with specimens with hantavirus, there's obviously some risk there, and I think that research is still very important But the political environment, I think, is making things really hard at this point. There's a good example. There's a bat vivarium that is being installed at Colorado State University right now, and it's getting a lot of political pushback. But for the things that we're hoping to do, the political environment, I think, is a little bit frightening. And I think the liability and sort of the lawyers and all involved at our institutions are looking at that and saying, oh, maybe we need to take a step back. And I'm not sure, uh, at the museum level, if we've really have dove off into that area of dealing with this new level of biosafety risk that we've, never really, I think, maybe done at this level. But also just the sort of dealing with, again, with the political environment that I think that we're now in at this moment in time. So whether or not the hantavirus approach in 2024, I'm not sure if it would be possible, to be honest, at least not without some serious um, coming together of very large parties. I mean, different federal agencies, large universities, museums. I mean, it definitely has to be a collaborative effort. I think the one-off institutional approach is going to be really hard to do going forward.
Yeah, great points, Cody. Thank you. It sounds like this is a pretty fruitful conversation, although I want to leave time for the other two parts. So please continue to share your thoughts on this in the chat. And there will be other opportunities. It sounds like this discussion is going to, it ties in a lot with part 3. So feel free to bring these things back up in part 3 too. But just for the sake of time, I'm going to move us to part 2 for now. Okay, so for this part, we're actually going to break out into 4 discussion breakout rooms. So in your breakout room, we will share a Google Doc where you'll keep notes. So either everyone keep notes in the breakout room or elect a recorder. We will have about 5 minutes in the breakout room, and then we'll come back together and synthesize our discussions. So during the breakout room, consider the scenario where we currently have a national weather service. How could we develop a national emerging zoonotic pathogen service that will issue Doppler radar warnings for locations or periods of time that are high risk for spillovers? We cannot sample everything everywhere all at once, so we must be strategic. How can we tackle a pathogen X scenario using an integrated One Health approach, inclusive of museums? And who would be involved? How would these assessments be made? What data would be needed? We'll copy these prompts into all of the breakout room brainstorming Google Doc note sections so that you have them there in your breakout rooms. And Jyotsna, thank you so much for correcting me. We have 10 minutes for breakouts. Can we separate into the breakout rooms now?
The breakout rooms have all been opened.
Okay, thanks so much. I hope you all had productive breakout room sessions. Ours, you know, we it was very fruitful, but 10 minutes was short. Yeah, the goal now is to spend the next few minutes synthesizing what you all talked about in your breakout rooms. So I think we'll just go through the breakout rooms and ask for your major topics of discussion for each of these 3 prompts. John, would you be able to share your screen of the prompts again?
I can do that. He says, what did I do with it? Oh my goodness. Did I lose the prompts? Joseph, can you help me out here? I may have just done something I didn't intend to do.
Oh, somebody else beat me to it.
Awesome, thank you.
Thanks. Okay, so group 1, tell us how you approach this strategic sampling conundrum in the context of emerging zoonoses.
All right, well, I was in group 1. So we talked about it from the perspective of what kind of centralization you'd need for data in order to pull something like this off, and recognize that, for instance, there are organizations like the CDC, which are in many ways tasked with things like this, but they're a pretty diverse organization, and how all that is pulled off at the CDC, I think, is open to constant discussion about whether or not they could do things in slightly better ways. And they have policies related to archiving material, but those are, and Marcy and folks should be talking about this. It's a hard thing to stay ahead of the game with respect to archiving samples for these kinds of types of projects.
Can you tell me more about that? You mean in terms of space, capacity, people, just getting those samples into collections?
This is Marcy, and Taylor can jump in, and I certainly speak from a very narrow lane, so I just want to put a disclaimer out there, but I think the typical activities at the agency is more reactive rather. There's so much going on, and you know, in the post-COVID world, we too are sort of in our infancy in trying to think differently about how we collect specimens. Again, mostly dealing with humans right now, which is, you know, different from my past. There's just a lot involved there when you're trying to collect samples that are not for a direct reason. I mean, you all go through this when you could sign your own consents. When they draw anything from you, you want to know what's going to happen to that sample and that data. And so to pose the idea that we're going to collect some of your blood and use it later for anything or nothing doesn't always pose well. So we're trying to establish now, you know, types of collections of samples that can be used sort of baseline for different purposes. But the kits that are involved, the assays that have to be developed for testing, there's nowhere out there. Most of you know this, I'm preaching to a very well-educated choir here, but there's not a general screening kit out there that would, I think, do the trick. And, you know, where that funding would come from and how it would be distributed are all things that are just kind of floating in my head, not even just thinking about our sample policies as they exist now. So I don't know, it's a huge nebulous thing to consider when you think about the agency and trying to do anything in a standardized way. I still think, unfortunately, at this time, it still works in a very pocketed fashion in trying to work with those groups that are specifically working on those pathogens of interest. I don't know, Taylor, if you wanted to add anything.
Yeah, Marcy, I think you covered most of what we talked about. We talked about some of the policies that CDC has in place, and that it's just a very large agency. And part of our responsibilities is to help make sure that those programs that are storing with us are following those policies and maintaining the data that they need to maintain, keeping the documentation that we expect for them to keep. So that was most of what we had talked about.
Yeah, I do want to point out one of the things I thought Taylor pointed out, which was really interesting, is that basically the biorepository is receiving things when projects are done. And, this gets into this kind of, for me, this concept of a specimen management plan is how do you get going before the project, as the project's starting. And of course, obviously the CDC is often responding to outbreaks as opposed to looking at things beforehand, but that would be one of the ideas in a perfect world of a forecasting institute for sure.
Great. I'm seeing some comments in the chat about the real-time versus benefits to sampling for real-time assessments and surveillance, and then the value of archiving samples. Jocelyn and Jennifer, did you want to bring up those comments here?
We can. I feel like we had talked about it a little bit in our group, but our group isn't presenting yet, or are we, Kelly?
Go ahead.
I think we talked about, Jennifer brought up a really awesome example of more real-time forecasting of pathogens similar to the National Weather Service for avian influenza. It was a great example because it is a little more real time, and it sounds like not only is it being regularly updated, but veterinarians and zookeepers are using this on a daily basis to determine whether animals should or should not be put in a situation where they may have potential exposure or not. And I think when Jennifer mentioned that resource, which I don't remember the name of it, Jennifer, could you pop it in the chat or feel free to interrupt?
Yeah, I'll see if I can find the website for that and put it in the chat.
Yeah, that'd be awesome. My response to that was, I think that's the dream. The challenge here is that we have all species and all pathogens to deal with. And so I think it starts becoming a little less manageable. We did talk also about the Pharos database, which I can also post a link in the chat. Pharos is kind of trying to do that for viruses. But one of the challenges is that none of the viruses that they're posting in real time are voucher backed or specimen backed. So it basically freezes host taxonomy and assumes it's correct in perpetuity. And so those data will quickly become out of date. But the idea behind Pharos, I think, has a lot of power to it. And I think we talked a lot about pipeline building. So it's not super realistic for any one organization, whether that's CDC or your biorepository or a genome center, to be responsible for all aspects of the pipeline that is needed to create a national weather service of zoonotic pathogens. Instead, what we're talking about here is building those pipelines and connecting those nodes, which I think we're doing by having these types of conversations. And so museums are kind of that boots on the ground surveillance, connecting them with other wildlife surveillance opportunities through veterinarians or wildlife work or medical surveys or outbreak response, getting those samples to genome centers to do that sequencing and bioinformatics, and then delivering that data and translating it in a way that is accessible to communities that can actually do things like public health announcements or things like that, because it's not going to be the biologists doing that. So that's where I think it's these teams of people and pipelines that are going to be key to this pie in the sky.
Yeah, great. That's you all did a lot in 10 minutes. That's yeah. Any comments or thoughts on Jocelyn's group's discussion? You all were group 3, group 4? Okay. Okay, so let's do group 2. Cody, do you want to, or Sheila, do you want to present for group 2?
Oh, do you want to get started? I am sharing my screen and I can't get to the notes.
Thanks, Sheila.
I will say that Vincent, who unfortunately had to leave us, mentioned the importance of just for him, for the CBP, the importance of communication and direct communication, because oftentimes communication, especially in terms of importing and exporting, it's secondhand or third hand. So it's often hard to track down the original source, which then creates problems in terms of permitting issue. So that was kind of one point that we spent some time discussing.
Yeah, our Cody has to step out really quickly, and our other group member Vincent had to leave the call. But yeah, Sheila, that's our group spent a lot of time talking about how one of the most fundamental shortcomings of our current system, or in our ability to achieve this type of forecasting center, is communication. Permits go for USDA, CDC, US Fish and Wildlife. We, as researchers, we submit them to these individual institutions, but then Customs and Border Protection are the ones who actually physically are holding our sample at the airport, trying to decide if we can get it into the country or not. So there's just these real-time breakdowns in communication or shared information that seem to be one of the biggest barriers to this. And it sounds like other groups are also talking about the importance of communication. For example, what group 4 brought up about the unified system for tracking influenza, avian influenza, across the US. Trying to look at our notes.
The other, just going off of memory, so the other thing that was mentioned is since we use National Weather System as an example of if we would mirror some sort of national emergent zoonotic pathogen system in the same way, where the state agency, especially in terms of border of things coming in, you would have local offices then kind of ping up an issue to the national branch and then have that sent out. We were kind of talking about it more in a, federal or state agency way, because it really, research-wise, we don't have the capacity to take on something that's so kind of using something that already has the infrastructure in place.
Yeah, and I'll add to that. I agree with that. That's I think one of the hardest parts is finding the central agency or institution to take the lead on that, to communicate out, this is what's happening, who's doing what. I did add into our notes, because we ran out of time, is, and maybe using some of these specimen, locators, repository locators, who has what samples already that exist in a specific area, so that may be archived, but then in a similar fashion, creating something that you can say, I am collectively working on collecting samples from this outbreak or whatever, that can then be communicated out. But again, I think the important part is finding someone centrally to be able to get all of that information and then get it out to the people that you need to get it out to. Even just sitting in on this meeting, I've heard of several other groups that are getting together, or, that have been mentioned, like the museums, what was that? Save the page. Museums and Emerging Pathogens program that I've never heard of before, because it's not in my wheelhouse. It's not a part of my daily job, but it could be something that my biorepository could provide, or samples from mine could provide information in helping with something if there's an outbreak or whatever. So I think just communicating out to all of these different areas, and like what Mackenzie said, the CDC is primarily human. That's one species. And as difficult as that is to work with that one species, when you're talking about the non-human samples, the environmental samples, you're talking about a plethora, hundreds of thousands of species. So you've got different connections everywhere. But yeah, communicating and connecting those dots is, I think, the important part. But who really takes the lead on that, and how do we get the support and the funding that's really needed? If it's going to be part of the federal government, which I'm a part of, it's getting the funding and that specific agency to provide the funding enough to be able to support that. And how do we do that when funding always seems to be cut every year?
Yeah, thank you, Rebecca.
Yeah.
I want to leave time for group 3 to tell us about their discussion.
So I agreed to be the spokesperson for group 3, but please, anyone else jump in if you'd like. And so we also picked avian influenza as our example. I think that's in everybody's minds these days. I'm personally getting roped into this situation right now. And so we had a discussion. Oh, should I share my screen with the notes? I can do that. That will work. Hopefully that's not too small. Okay, and so we had a discussion about how, if we had a system in place that had museums involved, where we had good flow of specimens, that might help in situations where we needed to go back and figure out, well, so how did flu jump from birds to cows, which we're still really not sure about. And so having cooperation between the different sectors, different stakeholders is key, and it can't really be reactive. It needs to be, there needs to be systems in place beforehand, I think. And we discussed, how would we get access to the necessary data to do mapping and risk mapping. And I guess I made the comment that as soon as a pathogen crosses over from the wildlife realm to the food production realm, things get a lot more politically difficult, at least in the US. That's definitely the case. And so we do have, various agencies that have certain data, but it's typically not made available without a lot of agreements in place. And in particular with our dairy industry, there are a lot of restrictions on that data, and it's very difficult to access. And so I think having partnerships with the states that are doing kind of ongoing surveillance is very valuable, and having kind of MOUs in place already on things such as, how would data get de-identified if they were publicly released. That all needs to kind of be figured out in advance. But at the end of the day, we have to be mindful of our industries and their bottom lines, and not hurting our economy in the process of trying to address these situations. And we also, we also had a comment about maybe potentially bringing in EPA so that we could deal with maybe less politically sensitive samples from the environment rather than from our agricultural animals.
Great. Yeah, thank you so much for presenting. Any other wrap-up thoughts before we move on to part 3? Okay, great. John, would you mind sharing your screen again?
Sure. Yep.
And then I'm going to turn it over to Greg and Joe to moderate part 3.
Are you talking, Joe, or am I talking?
You are talking, Greg.
I am talking right now. All right. For number 3, it is a lot of things we've already sort of hit upon. So right now there are all kinds of data. We know there are data in various places. How do we share those data with the people who need them? How do we either get the data to them or make sure that they know data exists and can go find it? Museums store a lot of digital data about the specimens. Sometimes that's called digital twin. And, basically we have, CT scan data or all kinds of data. Again, how do we share that? How do we make it fair? How do we integrate these data with existing initiatives? Very easy questions.
Well, Joe won't be surprised by this because I bring it up all the time, but with respect to relevant stakeholders, we've got to look towards the philanthropic community. And my pet peeve is that we can't get Bill Gates to buy into One Health type stuff in an effective way. And one of these days, that's actually going to happen.
Go for it. Laura's got her hand up.
I really feel like at least when it comes to the sequence data, NCBI is where it's at for sharing that data. I strongly agree that at least the sequence data should be public accessible, and I think NCBI is the best way to do that.
Sheila's making a point in chat. You want to say it verbally?
Oh yeah, I'm just saying, through our discussion, we use museum as a shorthand, but if anybody has thoughts on other types of collection and how, we can share these data, make it integrated with other initiatives, we're, very interested in hearing about them.
Yep.
Well, I'll just say Laura, I agree with you. And at the same time, one of the listening sessions was focused on genetic data and building those connections with the repositories like GenBank to, in real time, be able to actually correct and edit things from the perspective of what's known about the data beyond what the authors knew. And so identifications, for instance, in GenBank often change over time. And their policy currently is to only allow the people that submitted the data to be the ones that change it. And in a museum community where there are vouchered specimens, and as Sheila's saying, in any community like that where there might be people that actually know more about that specimen than the actual publishers of those data did, that could actually help work with NCBI to make it the database better on a broader basis.
John, I think the reality is a number of, or a few museum databases are tied directly to GenBank now, so that you toggle into an individual specimen. So if the specimen data have been updated or other things have been published on that particular specimen, that a researcher can find all that information. So there is a little bit of updating that does go on for sure if that direct toggle connection has been made. And, a few of our databases do that. And just one more riff on that. GenBank has published that their gold standard is a DNA sequence or sequence that is backed up by a physical specimen in a public repository, like a museum. And so this is really, the idea is to really make bulletproof, high quality data for science that, as Kelly pointed out in her first slide, is replicable and extendable.
Cody?
So one thing that, although I think those of us in the museum world would hang our hat on the quality of our data, or at least the presumed quality of our data, is that data does actually not ever get seen by, or rarely gets seen by, those in public health or working in biomedicine. And one reason for that is that we're the ones, of course, building our data aggregators as well. So those public front ends that are available to anybody on the web are built for people doing biodiversity science, and not necessarily those doing virology or or any other public health sort of application. And that's one thing I think that we could definitely do better on, and trying to take those data and kind of deconstruct them, or or deconstruct the portals to where you can enter them from a different perspective. So if you're interested in pathogen X, which we keep referring to, you at least could query a database from that perspective, dial down on potential host taxa that are of interest, figure out where samples are for those host taxa from a worldwide perspective, and be able to do your project by getting samples. I think that's one thing that we're that some of us at least have been talking about, but that it's reality. We need to Gates to pull up, or Jeff Bezos, or somebody to kind of make it happen. It would be relatively easy. I mean, we have the technical capacity within the museum community to do it. I think we just need to bring in additional folks from public health. Certainly there's people on this call that probably have a pretty good idea as to how that might be done.
Do you think we would need additional fields, or is it really just a matter of
I think flipping the data on its head, rather than querying by the mouse species, right? And you query by the pathogen, or at least or synonymize the names into more common names. For example, search by rats, mouse, bird, sparrow. I mean, something that's more inclusive and broader, but also could help sort of someone entering that may not have that biodiversity background or the taxonomic expertise to at least dive into those samples, find those samples, and then reach out to the different institution that may be holding them to figure out whether or not that's something that's applicable to what they're interested in.
Yeah. That sounds fun.
It does sound fun. And I think also, keeping track of how quickly samples are flash frozen and put in a stable environment from the field to the tube is something that I know when I get contacted by people in biomedicine, they want to know, like, how quickly was that tissue frozen. And oftentimes, it sat in a -20, and then it made it to a -80, and then it eventually made it into a liquid nitrogen.
Right.
That's not going to yield high quality RNA, of course, or if any RNA, let alone be able to detect those pathogens that may exist within those samples.
Let's do it. I think that would be complicated. I agree with you on the history of the tissue sample. I mean, I have a hard enough time knowing if something was found dead or collected, when it's handed to me from somebody. And that's it's a subtle difference, but it's an important difference. Like you said, how long was it dead before a sample was taken? Well, if you peeled it off the highway, I need to know that. All right. Yeah, DORs, exactly.
I think it's also important for biodiversity research as well. So I think the preservation, the way something's preserved, and then the way that condition changes through time. I mean, I wouldn't necessarily call myself a pathobiologist or even emerging infectious disease person, but I'm on a project right now where we need cryogenically preserved tissues, and they can't be held at -20 or -40. It has to be -80 or below, ideally in vapor phase nitrogen. And right now I'm really struggling in the informatics world, the biodiversity informatics world, because that information is very difficult to get from most collections. And there's a lot of really large collections that their procedure is to just contact them. And I'm looking for hundreds of individuals or hundreds of species. And that puts a ton of work on that institution to say, can you tell me the preservations of all of these that are not available online? So making that information available, while is a lot of work up front, hopefully removes some work on the back end. I think as we talk about developing this, I agree with everything Cody just said, but that's not surprising because I know Cody. But I kind of see two ways moving forward. There's the way where museums are kind of initiating this. We have the primary data and we are serving it in this form right now. We could serve it in another form. Or I see it in kind of the 1Z, 2Z direction that we're going right now, which is motivated by funding. So if there's funding for something like avian influenza, then a portal gets developed. I think the challenge with that is that frequently museums are left out of that development, or these biorepository sample archives are left out of that. If we can get integration across there, then I think 1Z, 2Z is a bit the way to get started, because you have funds to develop those fields for those pathogens in a way that connects to those communities. Otherwise, I think it puts a ton of new knowledge requirements coming out of museums to serve their data in as many different ways as there are pathogens, which I think is a little bit challenging. A lot of it challenging.
Yeah. Not as fun.
Less fun.
All right. Well, we've kept you now for getting close to 2 hours, so we're going to wrap up here. I want to thank everybody for participating. And I'm just going to finish up with a final slide here, which goes back to the kinds of things that we're thinking about with respect to conversations. We want to encourage you to continue thinking about this and go to the listening session web pages and put in questions or discussion topics that come up after this. I want to emphasize that there will be a final workshop. If you're interested in that, we'd love to hear that you are, and we'll set that up, and that's designed to be 50 to 60 folks with the BIOFAIR steering committee to go through some of these discussions. And again, the outcomes are going to be some recommendations that we think can move things forward, and we'll produce those in a couple of different ways. And if there are any last minute questions or anything I've missed in terms of housekeeping, why don't folks let me know? Anything? Well, folks, I'm going to give you 3 minutes at the end of this and just say thanks to everybody. I want to thank the key domain reps, Marcy, Kelly, and Joe, for being great. And it's been wonderful working with you to set this up. And again, thank you from the listening community. We really appreciate you coming on and talking about this. And we look forward to seeing where this will go. So thanks, everybody.